Welcome back to the course Fundamentals of General Immunology. So today we'll discuss something about receptor editing and also a little bit about the tolerance. We have looked into the B and T cell selections and also how these tolerance, central and peripheral tolerance are playing an important role in selecting specific B and T cell population. So in the last lecture we looked into that in the bone marrow and in the thymus, which are the central organ, where typically the B and T cell population which are reacting to the self antigens are getting cleared up. And these processes of clearing up is happening because either they are getting a clonal deletion or by a process of energy. So we'll look into those phenomena today in details and also try to look into some of the specific terms related to tolerance, although we will cover the tolerance a little later in this course, where we will discuss about mucosal tolerance, oral tolerance, and the stuff related to a non-immune response towards the gust epithelial layers. So we'll look into that a little later. So let's move on and try to understand this receptor editing. When we say receptor editing, it stands for the proteins which are expressed on the cell surface are getting edited in terms of there are changes which are incorporated into the amino acid levels so that the conformation of the protein can be changed. So for example, if there are alanine, phenylalanine or tyrosine or isolation, there are some set of amino acids which are present on the receptors of a protein. Probably post editing, those amino acids are changed, and because of this change in the amino acid, we see a whole bunch of different in the receptor biology when they are not binding to the cognate receptor. So this is basically a key immune tolerance mechanism where immature B cell, upon encountering a self antigen in the bone marrow or in the thymus, which we have developed. So these are especially the B cell which are encountering the self antigens. They are replaced by the autoreactive immunoglobulins which are reacting towards the self antigen. So basically, once you have a cell, and if this cell is expressing some receptor which are reacting to the self antigen, so it is going to mount an immune response in the future. So these immunoglobulins will be raised in order to fight against the self antigen, which is not required by the human body. So what is happening, there is an editing which are happening in terms of receptors. So now this receptor is changed. Because of this change in the receptor biology, now this antigen is no more able to bind, and therefore there is no immune response which is happening, and this is what you call receptor editing. So we will try to understand this receptor editing in terms of how this critically regulated. So these are actually non-reactive immunoglobulins rearrangement. So and then we can see that these are the processes which are decorated in a very large extent in the immune cells, particularly in the bone marrow and the thymus. So non-self-reactive ones through further light chain gene rearrangement, they are through to the process. So only the non-reactive species of the cells are getting through, otherwise they are all eliminated. This process is actually important because suppose if there are cells which are reacting to the self antigen, they have to be deleted out. Deletion process is actually an energy consuming process, and cells will lose a lot of energy in order to say make a new cell altogether, and therefore this is required that you can rescue the stuff, and to rescue the stuff it's better you can edit the receptor in the cell level so that it can have a say same cells can be utilized to do the process, and therefore it is called that it rescues cells from deletion, particularly the process of programmed cell death called apoptosis. It's also a quality control because it's a checkpoint that reshapes the antibody repertoires, and this is again very important because these all processes are critically controlled, and most of the processes are the ATP driven NGG process. So there is a requirement that one has to think about carefully that how these things can be minimized in order to process more and more antigens. So these are typically the change in the receptor biology which we are going to study here. So there are key aspects which are important in order to understand this receptor editing. although we have to recollect a little bit of the memory which we have discussed in our gene rearrangement when we discussed about the light and heavy chain in the antibody. So we discussed earlier that there are chains such as VDJ and they recombine it, right? And because of VDJ recombination, you get a variety of antibody, a variety of heavy and variety of light chains possibilities. Because of that, you see a different repertoire of antibodies that can bind to a separate and global varieties of antigen. And therefore, we need to understand the key mechanism which are involved here. So very important two molecules which we are going to discuss here is called RAG, and these are actually a recombination associated gene related to antibody gene shuffling. So we'll see it a little later. So there are two counterparts, RAG1 and RAG2. These are very specific genes which are involved in recombination process, VDJ recombination process, and how they do it, we'll try to see it in detail. So this often lead to the deletion of an existing light chain. What does it mean? That probably the kappa chain can be converted into lambda or vice versa. In order to change the receptor specificity, the light chain rearrangement happen, and because of the change in the receptor biology, we can always see that there are light chains which are produced differently. Again, this is a primarily way that autoreactive cells are getting eliminated. So these are getting silenced because of the process of receptor editing. This may happen that if there are cells in the immune system which are reacting to the self-antigen, there could be a receptor editing in order to induce a tolerance. So therefore, these are again inducing tolerance with the help of these two genes which we are going to discuss about, RAG1 and 2. In terms of location, we discussed this location little earlier also, that most of these phenomena are located either in the bone marrow or in the thymus. So the phenomena associated with the bone marrow, we call it central tolerance, and the one which is there in the tissue level, what we call as a peripheral tolerance. I'm repeating it again and again in order to emphasize the point that these two organs are critically controlling the receptor editing biology as well as clearance of the cells which are autoreactive in nature. And then we are discussing it in terms of clonal deletion and energy so that the cells can be further processed for other immune regulation. Well, if we are deviating anything on this, the human body may suffer from various complications or various disease conditions. So one such example is lupus, and this is actually an autoimmune disorder where the immune response are attacking their own cell population. So of course, the receptor editing does play a very critical role in maintaining the homeostasis of the host cell. Let's move on and try to understand the two genes which we are discussing in context of receptor editing. So the gene name is RAG1 and 2, which stands for recombination activating genes. So name itself indicates that there is a phenomena of recombination, and these are activating genes. So these are the genes which can activate the recombination. So the key aspect about again RAG1 and 2 genes are these are located specifically, and these are coded specifically in chromosome number 11, and we can call it 11p13, which is in the short arm, the upper arm of a chromosome. And this protein are basically containing core and non-core region necessary for the activity of this protein. So this essentially containing the core and non-core area. So these are the proteins which are acting as an enzymatic way in order to activate the recombination process. So these proteins are typically binding to a region in a gene, and these genes are called recombination signal sequences, RSS, right? We will see it a little later. So RAG proteins are produced by a typical coding sequence in a gene present in chromosome number 11, and these are specifically containing core and non-core region, and also these can bind very specifically to a region called recombination signal sequences that are flanking the VDJ sequence in the gene. So again, emphasizing the same point that we have gone through V, D and J chains in the light and heavy chain recombination event. So these sequences, the recombination signal sequence, are flanking these region. So suppose this is V, so these sequences are either flanking this side or this side. This is the area where the protein RAG can bind. So RAG docking area of RAG protein is basically a flanking sequence of the BDJ, which is also called as recombination signal sequences. These are expressed very specifically in the early developmental stages in the large concentration. So they are expressed more in the early stages, but they are also expressed later on in the life cycle when there is a requirement of editing of a receptor. So these are primarily on the T and B lymphocyte, and essentially if it is T and B lymphocyte, we can expect their location to be in the thymus or the bone marrow. So the expression of these genes are located in the thymus and bone marrow. So one should critically think about RAG1 and 2 genes separately as comparison to any other gene. So these are specifically genes involved in recombination event. These are expressed largely on T and B cell production area, which is thymus and bone marrow, and their production is largely in the fetal condition or embryonic condition, but they are also expressed later in the life in order to control the receptor editing and also avoiding the self-reacting T and B lymphocyte. Their functions, as I told, there are functions which are involved mainly on the receptor editing, apart from the initial maturation process of the B and T lymphocytes. This also a critical function apart from their own known function of maturation. So the clinical relevance is again when we are talking about any immune cells, any immune gene, any immune protein, any deviation in the structure or the function of a particular protein involved in any of the critical pathways in immune regulation can lead to disorder or disease or many other issues. Particularly in this case, there has been report that people may suffer from a typical kind of cancer called lymphomas. So lymphoma is a cancer of lymphatics or lymph node or lymph gland, where the proliferation of the lymphatics or lymph node is largely exaggerated in a way to give a stuff so that the person may start suffering from a disease called lymphoma. So lymphoma is actually a cancer of lymphatics or lymph gland. So we will move on and try to understand the sequences which we have discussed. So we discussed about recombination signal sequences, RSS. Now these are very critical, and we'll try to concentrate here and see that how these are happening in case of V, D and J chain. So typically these are suppose we will take D. So in case of D, these are the flanking sequence. These are called heptamer or nonamer. So there are genes which are the stretch of the 7 nucleotides here, ATGC combination, and or there could be a 9 nucleotides stretch. So these sequences are either 7 or 9, and then in between you have this 12 nucleotide spacer sequences. So these are the flanking sequences which are called heptamer or nonamer, and these are separated by this spacer sequence called either these spacer of 12 mer or the 23 mers. So just for the pictorial representation, we are showing here that in case of D, we are flanked by the 7, 12 and 9, but in case of V, we are flanked here by 7, 23 and 9. And here also in J chain, 23 is 7, and so here it is 7 and 7. So that means we have the flanking sequences of the spacer, and this is the spacer region where we can see the docking of the RAG protein. So this the spacer sequence are important, and there is a rule called 12-23 rules. So what it says that the 12 can only bind to 23 and not with 12. So that means the 23 to 23 combination is not possible, 12 to 12 combination is not possible, right? So the only possibility which exists is 12 to 23. So that means this is what is rule 12 to 23 rule. So a gene segment flanked by a 23 specific recombination signal sequence can only be linked to a segment flanked by the 12 mer recombination specific or signal sequences. That means only the combination which are possible is between the 23 and the 12 and not between the 12-12 or 13-13. So this is very important rules which exist in the B and T lymphocyte rearrangement when it comes to the RAG protein. So just keep in mind that the genes are actually flanked by heptamer or nonamer with a spacer of 12 and 23, and 12 and 23 can bind in a way so as to make divergent or receptor editing, but the phenomena is not possible when they are flanking with the same homologous sequences such as 12-12 and 23-23. So let's move on, see how this is happening in context of a B cell or T cell receptor area. So here is the biology of the RAG1 and RAG2 in context of a gene. So let's try to see here. So the RAG protein complex bind to 12 and 23 base pair sequences, spaced recombination signal sequences. So these are the RAG proteins. So this is one RAG, this is the other RAG, and these are the two flanking sequences which needs to be recombined. So the protein complex bind to each other. So then they are coming, so they are forming a closed loop like structure so that both the proteins can come close to each other. And once they are coming close to each other, then they make a stagger cut, first a single strand and then double strand. And once there is a cut, so this DNA double strand cleavage can make a hairpin like structure at the end of this gene segment, and then these two fragments which are carrying the RAG protein are getting segregated out. There are many other proteins which does play a very important role, and some of them are listed here. One is called Ku protein, and the 70 and 80 Ku is the one which is important, that play a role during this process, and also DNA dependent protein kinases, because these are required to do a process of ligation and other important process which requires the joining of these two chains together. So therefore we need a ligase enzyme as well. So these hairpin structure are joined following the cleavage of the RSS end. Following that, we need to understand that to progress more of the heterogeneity in this chain, we have seen the enzyme kinetics of TDT, terminal deoxynucleotide transferase, and here also the terminal deoxynucleotide transferases are coming into picture, and they add more heterogeneity to the population and leading to more and more incorporation of changes which can lead to receptor divergence in the chain. And finally, the DNA ligase that comes into picture, which can ligate the coding region in order to code for a protein, and of course the signal sequence also join, and then they can segregate out. There are proteins which are involved here called XRCC4, which is called X-ray repair cross complementary protein 4, which is an essential component, and essentially what we are discussing here is called non-homologous end joining, NHEJ, so which we'll study in molecular biology, or we have studied that thing in molecular biology. And also here, since the two ends are not homologous, therefore we call it non-homologous, because these sequences and these sequences are actually different in nature, and because of different nature we called it non-homologous end joining, because the end to end of the genes are getting joined because of the DNA ligase present here, and this protein does play a very important role here. So what is emphasized here is we understood the 12 and 23 rules. So there are 12 and 23 sequences, spacer sequences which can bind to each other. The RAG protein come into that site, they can make a hairpin like structure which we have discussed here, and after making a binding to this region there is a cleavage occurring, and the cleavage can lead to the first strand cut and then double strand cut. Following that, there are incorporation of a lot of proteins such as Ku 70, 80 and also other kinases which are present there. And finally, there are more divergences which are added because of the terminal deoxynucleotide transferase, which are adding more heterogeneity to the gene. And finally, the ligase come into picture, which can join these two coding sequences which came close to each other because of the RAG protein, and then leading to a very different kind of gene altogether. So the whole process could lead to divergence in the receptor editing, and the major player here is RAG1 and 2 proteins. So we'll move on and try to see what else is required in order to understand the receptor editing. So what we have understood is the two ways we are trying to see the divergence in the B and T cell. One is called clonal deletion, where the cells are dying, where upon the receptor, if they are undergoing receptor editing, they are surviving. If they are not undergoing receptor editing, they are simply getting deleted by apoptosis. And if at all not, then there is another process that exists in there, which is called energy. Now energy is a process where the cell become non-functional without any death. So death is not happening. So we could see this in context of three different ways. There could be death of the cell if there is a reaction. There could be the editing of the receptor without any death, so that the binding can be aborted with the self antigen. And the third, if at all the cells are reacting, and if the body won't want to waste the cell in order to make them apoptotic cell death, they can simply do a functional-less cell. So the cell won't be functional, and the condition is called energy. So we'll try to understand what do you mean by energy. So energy is actually a complex process where the cyclic process, the whole cell become non-functional in terms of the energy driven pathway. So let's try to understand this in context of a normal cell. So in the normal cell, the cell membrane, the plasma membrane, contains multiple proteins, and one such protein here is called PIP2, which stands for phosphatidyl inositol 4,5-bisphosphate, which is a critical membrane protein which can be cleaved into two fragments called inositol phosphate, triphosphate, and also diacylglycerol with the help of this enzyme called phospholipase C. That means this PIP2 is a membrane anchoring protein. Upon binding to phospholipase C, it gets cleaved into inositol triphosphate and diacylglycerol. Diacylglycerol is actually a very important molecule which activates a MAP kinase pathway. And once this cleavage can happen, then this RAS-GRP pathway, which is actually a RAS guanine nucleotide releasing protein pathway that can sense and activate RAS protein by exchanging the GDP and GTP conversion, right? So essentially we can see that these guanine nucleotide releasing protein can do a biology so that the cell can be undergoing MAP kinase pathway, and therefore the cell become activated. That means any external stimuli that comes into this T cell can activate the T cell by this pathway, which is kind of a very well documented pathway. Now inositol triphosphate is actually the calcium mobilizing pathway. So the calcium is getting mobilized from the cell, which in turn do again a lot of other pathway which can activate the T cell by downstream signaling activation. Now this is happening when the cells are getting activated. What will happen if there is an energy? Energy process will avoid the activation of T cell, and how it does that is because the PIP2 protein can cleave into IP3 and diacylglycerol. And diacylglycerol is actually worked upon by a very specific protein called DGK, which stands for diacylglycerol kinase. Now diacylglycerol kinase can convert this DAG into phosphatidic acid, and because of this conversion of phosphatidic acid, now this DAG is no more able to activate this pathway. So this pathway is not activated simply because this diacylglycerol specific kinase is getting activated. That means the whole signaling is defective in this particular cell, and because of that region, this particular cell is not getting activated, and the condition is called energy. Now the energy is simply a non-activation of the cell. Cells are still surviving, these cells are non-reactive in nature, and therefore we won't see this cell to be participating in the immune system in general, and therefore this is also a kind of a homeostatic mechanism adapted by the host cell in order to stop the activation of T cell, while the pathway which we listed here, the PIP2 kinase pathway, is actually leading to activation of T cell through the calcium metabolic pathway. We could see that there are a lot of other T cell activation pathway which we'll see little later, such as NF-AT, another pathway that could lead to activation of T cell, gradually leading to high immune response. So this is all about looking into the receptor editing and how the receptor is involved there. One more term which are listed in this topic is tolerance, and we have gone through the central tolerance and peripheral tolerance, and again other kind of tolerance we will discuss a little later. But one important tolerance is, we'll just like to emphasize here, is called feto-maternal tolerance. Feto-maternal tolerance stands for the fetus kind of a tolerant in the mother uterus. So we know that the fetus are developed into the maternal uterus for a period of gestation, which is 9 months in case of human beings. So how this fetus, which is supposed to be the external antigen, is residing into mother body without getting rejected. And in immunology, the birth of a baby is actually immunological rejection. So once the body understands that the baby, fully grown baby, is now an external agent, then the body can throw it out, which is what is actually leading to the birth of a baby. So till the gestation period, the fetus can be surviving in the uterus because of this very important phenomena called feto-maternal tolerance. So this prevents the maternal immune response against the developing fetus. Otherwise, we could see that if this phenomena is not happening, then there could be an abortion in the first trimester or the second trimester in case of females. And the very important aspect about all this phenomena is the level of placenta and the involvement of placenta. So the placenta reduces the exposure of the fetus maternal immune cell. So the immune cells which are circulated in the fetus are not getting mixed with the mother side because of the barrier, and this barrier is actually placenta. And again, the outer layer, the protein expressed on the outer layer of the placenta may limit the recognition and act as a suppressive signal for the fetus to survive there. So what we understood here is, since the birth of a child is actually a synchronous process, and initially the fetus needs to survive, so we really want a uterine system to be non-reactive towards the fetus. Otherwise there will be an immune response and the fetus may be coming out from the mother uterus, so which can lead to abortion. And therefore this tolerance mechanism is playing very important role in the uterus of a female in the early stages, and the very important cells which are taking part in this process is placenta. So the placenta can do a barrier, can reduce the exposure of the immune cell transportation between the fetus and the maternal body, and also this expresses a lot of protein on the cell surface which can reduce the recognition of the immune cell from the mother side, and also it can lead to suppression of lot of pathways which can allow the fetus to survive there, which is not happening at the last stage. And once this is not recognized, the placenta become thin and not able to tolerate this phenomena or this kind of tolerance, then we can see that immune rejection is happening, which is what we learn as childbirth. So this is what is very important in terms of tolerance, which we need to understand. And we'll try to look into some other phenomena in our next topic. Thank you.